Welcome students to a short lecture about the two physio-X activities that are associated with the respiratory and renal system unit. That is Physio-X 9 about renal system physiology and Physio-X 10, which is acid-base balance, so regulating our blood pH. We're going to start with renal system physiology, Physio-X 9. One of the first activities that you're going to look at is the formation of urine. In particular, you're going to focus on the first step in making urine, which is filtration, right? The blood comes into the kidney, and the glomerulus here actually acts like a sieve and filters out waste products and everything else out of the blood. And then, of course, the other two kind of processes are reabsorption, so anything that gets filtered but is useful gets reabsorbed by the body, and things that are too big typically to be filtered but are still waste products will be secreted by the tubules. But the Physio-X activities focus primarily on the filtration process, because it is, of course, that kind of foundational step. So this is the glomerulus here. You can see it's this little capillary bed surrounded by the Bowman's capsule. The glomerulus is really different from other capillaries in the body in that it is both fed and drained by arterioles, right? Like your standard capillary gets fed by an arteriole and then drained by a venule. But the glomerulus is different because both kind of the feeding and draining blood vessels for this particular capillary bed are arterioles. They're both high resistance vessels. So that makes the glomerulus a very high pressure capillary bed compared to like your standard capillary. The other thing that's really different is that the afferent arteriole leading into the glomerulus has a larger diameter than the efferent arteriole leading out of the glomerulus. That essentially creates like a backup pressure, more fluid coming in with a smaller space to leave out. Again, imagine like 3 lanes of traffic trying to merge into one. That creates like a back pressure. So that also contributes to this very, very high blood pressure in the glomerulus. And it is that high blood pressure, that hydrostatic pressure, the pressure of the fluid in the blood that pushes fluids and solutes out of the capillary and into the surrounding capsular space. Now, there are also 2 inward forces. As the capsular space fills with fluid, that fluid also exerts a hydrostatic pressure, pushing fluids and solutes back into the capillary. And then the plasma proteins, which are generally too large to be filtered, they remain in the blood inside the capillary, and they exert a pull that pulls fluids and solutes kind of back into the glomerulus. But those 2 inward forces are not enough to overcome that very, very large glomerular blood pressure as an outward force, and we still will get kind of a net outward pressure and filtration occurring. So if we change, since the diameter of the arterioles are kind of one of the contributing factors to this very large kind of outward pushing force, if we change the diameter of those arterioles, then we can change the filtration pressures. And that's actually one of the activities that you will do. You'll get a chance to kind of change the diameter of the afferent arteriole and the efferent arteriole, and also kind of see how they change in response to systemic blood pressure changes, and kind of see what that does to the rate of filtration, how it changes depending on systemic blood pressure and the different diameters of the 2 arterioles. Another activity in Physio-X 9 deals with the transport maxima. So most of the things, or a good chunk of the things that we reabsorb out of our filtrate that are useful for our body that we want to keep, and a large portion of them, particularly like big things, when they get reabsorbed in the renal tubules, it's an active transport process. And that active transport process requires carrier proteins, transport proteins. And so practically everything that we reabsorb has a transport maximum. This is like the total number of a particular substance that our body can reabsorb. And it really is a reflection of the number of carrier proteins available in the tubules, right? So hyperglycemia is a really good example. High blood sugar, right? So if you have really, really high blood sugar, then when the glomerulus filters your blood, you're going to end up with very high levels of sugar in the filtrate. Well, there are only X number of kind of carrier proteins available to reabsorb the glucose, and so your body will reabsorb glucose until all of the transport proteins are in use. That is then our transport maximum. And at that point, the reabsorption of the glucose will stop and glucose will be excreted in our urine. This is why you can use urinalysis tests to make inferences about what's going on in the body. Right? If glucose is something that is normally reabsorbed and it's showing up in your urine, it means that your blood sugar had to be really high to begin with because your body couldn't reabsorb all of it. The other activities in Physio X9 deal with how hormones affect the renal system. And the two hormones in particular that we're going to look at are ADH and aldosterone. And both of these we've seen before when we looked at the endocrine system, but now we're going to kind of look at them from that renal perspective. So to remind ourselves, ADH is a hormone that is made by the hypothalamus and then stored in the posterior pituitary. And ADH's job is to tell your collecting ducts in your nephrons to reabsorb more water, typically to prevent dehydration. So when ADH is not present, you'll see the collecting ducts are essentially impermeable to water. So you end up with a very large volume of fairly dilute urine, right? This is the situation where you're inside in a temperature-controlled environment, you've had lots to drink, and your body isn't trying to conserve water, and so you end up with a very large volume of kind of pale yellow urine. Now imagine a different scenario. You're outside in Georgia, and it's 95 degrees, and you're sweating bullets because it's 95 degrees with crazy humidity. So in that instance, your body is losing water through your sweat. So that increases kind of the saltiness of your bodily fluids, and that is what triggers the posterior pituitary to release ADH. And when ADH is released, then it targets the collecting ducts here and puts what we call aquaporins, which is just a fancy word for literally water pore, into the collecting duct, and makes the collecting duct very, very permeable to water. And so the collecting ducts reabsorb more of the water out of your filtrate, and you end up with a very small volume of urine, and it's also very concentrated, right? It's very, very dark. And that really is kind of the role of ADH. Now, aldosterone also results in water reabsorption, but its triggers are a little different and its targets are a little different. Aldosterone is made by the adrenal cortex, typically in response to low blood pressure. So if, for instance, you have a low blood pressure for whatever reason, low heart rate, hemorrhaging, blood loss, high levels of vasodilation in your blood vessels. If your blood pressure decreases for whatever reason, that will lead to aldosterone being released from the adrenal cortex. Now, aldosterone actually targets the distal convoluted tubule part of your nephron, and it really causes the distal convoluted tubule to reabsorb sodium, but sodium exerts kind of a, kind of like a tag-along pressure on water, because water tends to follow sodium wherever sodium goes. So since aldosterone causes the distal convoluted tubule to reabsorb a lot of sodium, water kind of comes with that sodium, and you end up reabsorbing a lot of water. That increases your blood volume, which then increases your blood pressure. And that's really the first activity, the Physio X9 activity. The Physio X10 activity is all about regulating our blood pH. If you'll recall, our normal blood pH is about 7.4. So we have kind of slightly basic blood. And if we are in that range, 7.35 to 7.45, that is where our body functions best in terms of kind of proteins functioning, everything kind of being in those kind of homeostatic ranges. When we get out of that very, very narrow range, if we become less than that, we get into an acidotic situation. We become more than that, we get into an alkalotic situation. And you can see it's really, really not a wide range. If your alkalosis gets really bad, or if your acidosis gets really bad, it's not only like damage, it can actually be fatal. So we really do need to keep our blood pH in a very, very narrow range. And part of the reason that we have to have regulatory mechanisms for pH is that the hydrogen ions, of which pH is a measure, is a byproduct of our body's metabolism. So when we do aerobic respiration, part of that process is, of course, ATP, so we can power our body, but also carbon dioxide. And so carbon dioxide is a byproduct of our body's metabolism. Well, carbon dioxide combines with all of the water in our bodily fluids to become carbonic acid. And then carbonic acid dissociates into hydrogen ions, which are acidic, and bicarbonate ions, which are basic. And so just the act of our body's metabolism making energy, which of course we have to do, produces hydrogen ions, which changes our blood pH. So we have to have buffers. We have to have ways of regulating our blood pH. One of the ways that we regulate our blood pH is with the use of chemical buffers. Now, chemical buffers are sets of chemicals in the body, different molecules that resist changes in pH. They can act very, very quickly, so that if blood pH kind of swings one way or the other, the buffer's kind of right there to do its job and kind of prevent that change from becoming too extreme. The best chemical buffers are a mixture of a weak acid and a weak base. The base gets to counteract any increases in acidity, and the acid gets to counteract any increase in alkalinity. The weak bases and weak acids work best because they don't completely disassociate. Strong acid, something like hydrochloric acid, which will completely disassociate into its constituent ions, don't work as buffers because they just contribute to the problem. So these mixtures of weak acids and weak bases, because they don't disassociate completely, are really kind of the best chemical buffers. Now, you have 3 different chemical buffer systems in your body. One of them uses phosphates. One of them actually uses the proteins that we find in our body, in our body's cells and in the blood plasma, and so on and so forth. But the best, kind of most important of the chemical buffer systems is the bicarbonate buffer system. So carbonic acid is a weak acid. Bicarbonate ions are weak bases. And so if the blood pH becomes too basic, then carbonic acid can be a hydrogen ion donor, which will release hydrogen ions and bring the blood pH back down. It'll make it more acidic. Bicarbonate ions, if the blood becomes too acidic, get to act as a hydrogen ion acceptor. They can take hydrogen ions out of solution, which would then bring your pH back up. And so this buffering system is incredibly vital to keeping us in that kind of normal range. However, if things get out of overwhelmed, the chemical buffer systems, then we use physiological buffer systems. And they regulate pH by essentially controlling what we excrete. The lungs will allow us to excrete carbon dioxide, that changes our blood pH, or hold on to carbon dioxide. By changing our excretion of carbon dioxide, we can change our blood pH. And then the renal system allows us to excrete bicarbonate ions, hydrogen ions, maintain hydrogen ions, bicarbonate ions. And so we can really change our blood pH there by changing what we lose through our urine. Now, the respiratory system tends to respond faster. If your blood pH swings out of whack, within minutes, your respiratory system will kind of change its functioning to regulate blood pH. But it can only do so if the swings in pH aren't that bad. The renal system takes a little bit longer to kick on because we're talking about changing what we reabsorb and what we secrete. So it takes a little longer, maybe hours, a couple days, but it can handle wider swings in pH, and so it has a larger impact. But by controlling what we excrete, either as carbon dioxide exhale or things lost in our urine, these two systems help us regulate our pH and keep us within those homeostatic norms. So we're going to talk about when the respiratory system screws up first, and then we'll talk about kind of when the body screws up and how the respiratory system can respond. So if we talk about respiratory alkalosis, usually this is that we are hyperventilating, right? When we hyperventilate, we're exhaling a lot of carbon dioxide. And so we're actually exhaling more carbon dioxide than we're producing. And so we have a decrease in hydrogen ion concentrations. And so our pH goes up. Our blood becomes too basic. Well, in that instance, if our blood is too basic, we have to get rid of the base and we have to keep the acid. So we will excrete bicarbonate ions, which are basic, and we'll reabsorb lots of hydrogen ions because they're acidic. That will bring our pH back down to normal. If we have respiratory acidosis, this is the exact opposite problem, right? This is what occurs when you rebreathe. So when you guys did that rebreathing activity, if you had continued that rebreathing activity for a longer period of time, you could have gone into an acidotic situation. Or if you have any sort of hypoventilation, if you have an airway obstruction, if your respiratory center in your brainstem is inhibited, or if you have COPD. All of those things result in too much carbon dioxide in the blood, which drives down your pH. And so our blood becomes too acidic. In that case, we get rid of the acid, keep the base. So we will secrete the hydrogen ions and make sure that we reabsorb the bicarbonate ions. And so that's how the renal system can respond if the respiratory system screws up. Then we can have ways that are a whole host of different kind of ways and different causes in terms of a pH imbalance that roughly fall into what we call metabolic imbalances. So we'll look at alkalosis first. Again, too basic. So there are lots of different ways that the blood can become too basic. That isn't just a respiratory system screw up. If you take a lot of antacids, that can drive up your pH. If you are ill and you are vomiting, which causes a loss of hydrogen ions, right? So if you lose a lot of stomach acid, that's like kind of that burning feeling you feel when you're vomiting. Or if you're constipated. Because when we're constipated, we have a lot, the feces contain a lot of bicarbonate ions, and since we're retaining them, that can drive up our blood pH. Or if you have just a decrease in metabolic rate, let's say you have a slow metabolism, that means less carbon dioxide is being produced, which could drive up your blood pH. When that happens, one of the ways that the respiratory system can respond is by essentially slowing down the rate at which we breathe, or making our breaths more shallow so that we retain carbon dioxide. If we're retaining carbon dioxide becomes carbonic acid, which dissociates as hydrogen ions, and we can our blood can become more acidic that way to kind of counteract the alkalosis caused by any of these causes. Acidosis occurs when we're losing bicarbonate ions and the blood is becoming too acidic. Ketoacidosis is definitely one of the ways we can end up with acidosis. So people who have diabetes mellitus, the blood sugar diabetes, if their sugar levels get too high, the sugars convert into something called ketoacids. Well, ketoacids are acidic. Ketoacidosis, if untreated, can actually put you in a coma. But it can lead to an acidotic situation. Salicylic acid poisoning. If you take too many aspirin, that can lead to metabolic acidosis. If you have diarrhea, you're losing then lots of bicarbonate ions in your feces. Or if you have too much alcohol. If you've ever heard like horror stories about somebody getting alcohol poisoning, part of that alcohol poisoning is this, metabolic acidosis. Alcohol in the body is broken down into acetic acid. Acetic acid is, well, acidic. It will drive down your blood pH, and that is one of the things that is so damaging about too much alcohol. Another way you can end up with metabolic acidosis, which seems somewhat counterintuitive, is if you do too much exercise, right? Normally everyone's like, oh, you have to exercise. But actually, if you do too much exercise, you can end up with a buildup of lactic acid, which will drive down your blood pH. And then lastly, just an increase in your metabolic rate, right? Let's say you have an overactive thyroid. If you have an overactive thyroid, that thyroid is going to be producing lots of thyroid hormone, which is going to kind of jack up your basal metabolic rate, and your body's going to produce more carbon dioxide. More carbon dioxide, more acidity. In terms of compensation, one of the things that the respiratory system will do is it will hyperventilate. You will increase the rate and depth of your breathing in order to expel kind of the kind of quote-unquote excess carbon dioxide, which will bring your pH back to normal. Maintaining blood pH is really an act of those chemical buffering systems, but really focusing on the respiratory and renal systems. Without these two excretory systems, our blood pH would be much harder to maintain.